This fourth video on the linkage chapter will focus on the mechanism of crossing over or recombination. This is actually found in chapter 12. It is not found in the linkage chapter, so you can find it in your textbook on the pages indicated. So if you remember, crossing over occurs during meiosis 1 in prophase 1, and crossing over occurs during the formation of these tetrads. It's pretty interesting. There is always at least one crossover for every chromosome in meiosis. Your textbook goes into two different mechanisms. We're only going to talk about the double-strand break model. The other model, called the Holliday model, is really, I think, thought to be an incorrect model at this point. So the double-strand break model has the most evidence supporting it, and so that's the one we're going to learn. So let me remind you that crossing over occurs between non-sister chromatids. The way this happens is shown here. So we've got two non-sister chromatids, and the blue one, these two lines are just the two strands of that double helix DNA, and the other non-sister then would be shown in red down here. So the stepwise thing that happens is, first of all, one of the strands gets a double-stranded cut in the DNA. You get this cut, and then the ends are actually trimmed back. After end trimming, the single-stranded DNA gets covered by these particular proteins, which give this single strand sort of the power to invade the other non-sister chromatid. And what happens next is they start to look for a similar sequence. This structure at this point would be called a D loop because it kind of looks like a D if you turn it on the side. Okay, I'd like to switch now to another figure. So let me just review those steps that I just spoke about in the previous figure. So first of all, we have that double-stranded cut. We get end trimming on that top blue strand, and then one of the strands can invade the other non-sister chromatid and start to look for that match. Once it finds that match, then it's going to start to displace that second red strand of the DNA. And so here, this strand gets displaced, and we actually start to form additional DNA sequence using DNA polymerase. Okay, so this bottom one is used as a template, and then this top one is actually synthesized. At some point, these two strands hook up again, and so this one will actually get reattached up here. At the same time, this other one that was cut back is going to use this displaced red strand as a template to synthesize a new strand. And so here, this dotted line is a new strand, and then this one eventually will get attached onto this one. Now, in the process, if we step back a little bit and think about what's happening here, we've essentially switched the strand that we are base pairing with in this middle portion, right? So normally the blue would be pairing with the blue, the red would be pairing with the red, and these two middle ones have actually switched who they're base pairing with. The structure at this point is called a double Holliday junction. And so here is one Holliday junction, and here would be another Holliday junction. Another way to depict that Holliday junction is actually shown here. And this looks a lot like the chiasmata that are observed during the actual crossing over event. So in order to continue on with meiosis, in order to separate the homologous chromosomes in anaphase, that Holliday junction must be resolved. This resolution is done by actually snipping the DNA strands. And so there are a couple of ways that this can actually happen. We can actually snip DNA in one direction or in the other direction. One of those snips would lead to a crossover event. The other one would lead to a non-crossover. In class, we're going to look at some of these videos to just kind of review and look at some animations of the steps, which I think help a lot in making it a little bit more clear.